first unit is the introduction to bioinformatics applications
sir.
and database management. Second unit, genome analysis, so molecular biology and bioinformatics, and central dogma theory, genome tools. So how to declare the data from the web search, then both unit. So sequence alignment, then method of sequence alignment, multiple sequence alignment, then 15 unit protein structure prediction, primary structure, secondary structure, can go and find with the structure prediction model. So this is the overview of bioinformatics paper. It's a basic level. Okay. So what is the bioinformatics? So where we can able to apply the tools. Okay. Then application oriented. So now we are going to see the introduction. So introduction. So what is the bioinformatics? So bioinformatics is structural based studies of genomes. Okay, structural based studies of genomes. The development of genetic engineering technique is a big step forward in our understanding of genetic constitution of living things to enhance the medical and industrial capabilities. Advances in molecular biology in the early 60s were largely due to the academic interest, commitment to the development of knowledge. 1970s, the direction changed from the academic to commercial. This was owing the emergence of recombinant DNA technology or genetic engineering. So, the genetic engineering and recombinant DNA technology bloom another. So, normally in laboratory method, we had seen in the molecules in or gel based or quantitative based, qualitative based, and the method we can able to analyze the any kind of molecules. Okay, so after blooming the bioinformatic tools, so softwares and the genome prediction tools, the structure prediction tools developed from the computer technology. So after that, it will slowly blooming for the technology upgradation of bioinformatics. Okay, so the information retrieval from the biological database. Bioinformatics is nothing much the information retrieval from the biological database. So first we know that genome. So after the genome, it will transcription process. It's a transcriptome. Okay, gene means the coding for the molecules. Genome means it's a structural. OME, it's a structural molecules. Structural molecules. So bioinformatics. So proteomics, genomics, proteomics, it's a study of protein molecules. Genomics, it's a study of genome, genetics. Okay, metabolomics, it's a study of metabolic pathways. And the OME, it's coding for the bioinformatics tools. So genome, gene, it will convert the translation process, then protein synthesis. Protein synthesis. So after the protein synthesis, the protein going for the metabolic reactions, so inhibition, activation of the metabolism, so targeting of the protein. So again, those molecules, it will going for the genome. This is a cyclic process for the central dogma of theory. So molecular biology. So this is a concept. The people are studying the bioinformatics. So bioinformatics is a collection of the study in molecules. Okay. So overall studies of the genomic level and proteomic level, on transcriptomic level, or metabolomic level. Okay. Pharmacogenetics, கைனடிக்ஸ் அந்த மாதிரி. ஸோ எங்கெல்லாம் வந்து ஸ்ட்ரக்சரல் ஸ்டடீஸ் எல்லாம் வந்து லிவிங் திங்ஸ்ல பண்ண முடியுமோ அங்கெல்லாம் வந்து வி கேன் ஏபிள் டு இம்ப்ளிமெண்ட் தி பயோஇன்ஃபர்மேட்டிக் டூல்ஸ். So usually first the people are launching the human genome project. There is a successful completion of the human genome project in April 2003. They are starting from 1986. So 86 la start பண்ணாங்க human genome project. They will complete the entire human genome in the April 2003. Okay. So they have a different kind of milestones in the couple of decades. Okay. So the human genome project is the complete genetic map of man. Complete genetic map of man that defined by the restriction map fragment length polymorphism. Restriction map fragment length polymorphism. So RFLP, restriction fragment length polymorphism. So whole genome become able to sequence in one way. Okay. So a single reaction we can able to sequence in the whole genome. So we should fragment it, restricted, fragmented. So we're using the restriction enzyme use panni, we can able to fragment it our whole genome. So whole genome on the fragment panni, the people are studying those fragments. The complete sequencing of all the complementary DNA in man. So complete complementary DNA. So single strand synthesis ஆகும். அந்த single strand synthesis ஆகிறதுக்கு complementary strands, opposite strand full study பண்ணிட்டாங்க. Then identification of heterogeneous gene causing the genetic disorders. So அதுல வந்து எந்த genes எல்லாம் வந்து நமக்கு வந்து disease cause பண்ணுதோ அந்த genes எல்லாமே they will predict. Okay. So the existing information itself estimated there are 30,000 genes in human genome. அதாவது வந்து கிட்டத்தட்ட 30,000 genes வந்து நம்மளுடைய ஜீனோம்ல இருக்கு. ஸோ நியர்லி பார்த்தீங்கன்னா 1.5 million proteins, 1.5 million proteins available in our body system. ஸோ பார்த்துங்க எவ்வளோ பெரிய genome. So 30,000 genes. Can I able to see slide 3?
Slide 3, sir.
Yes, sir.
Okay. So 30,000 genes la vandu we have a 1.5 millions of proteins available. So approximately 300 tetrabytes files. எவ்வளோ ஃபைல்ஸ் இருக்கு நம்மளோட ஜீனோம்ல? 300 tetrabytes ஃபைல்ஸ் இருக்கு. ஸோ அவ்வளோ பெரிய ஜீனோம் வந்து they have completed the sequencing process. So network issue. So system wires and the system like அதனால மொபைல்ல சிஸ்டம்ல வந்து பிபிடியும் ஷேர் பண்ணிருக்கேன். Yeah, will connect. Yeah. Now you can able to see the okay. So human genome project sequence more than 1 lakh genes encoded for the 10 into the 10 power 3 into 10⁻⁹ bases. Okay. The other genome, Haemophilus influenzae, Saccharomyces cerevisiae, Drosophila, then Cytosoma, Plasmodium have also increased in the information from our web pages. So இது எல்லாமே வந்து நிறைய nowadays the people are completed more than number of genes, number of sequences have completed from plant, animal, bacteria, insects, rodents. So எல்லாத்துக்குமே வந்து full genome வந்து the people have completed. The first significant macromolecular sequence database was created by the Dayhoff, the form of atlas of protein sequence and structure, which has generated a number of databases all over the world. First, and the database sequence databases on the create பண்ணது பாத்தீங்கன்னா Dayhoff. For the macromolecules, it's the protein molecules. Protein molecules தான் வந்து first database create பண்ணாங்க. So nowadays the number of databases available in GenBank. So GenBank அப்படின்னு பார்த்துட்டீங்கன்னா sequencing bank. So நம்மளுடைய DNA நம்ம சீக்வன்ஸ் பண்ணக்கூடிய DNAs எல்லாமே வந்து you are going to keep it in the GenBank. Then EMBL, then PAR, Protein Information Retrieval Tool, PAR. Swiss Protein is the Swiss Sweden-based protein database. OMIM, then ProCite, Medline, PubMed. So இதெல்லாமே பார்த்தீங்கன்னா it's a databases. Okay. So the store of sequence information on nucleic acid, protein, restriction enzyme, cloning vector, transcription factors, the storing information on cell line, genetic disorder in humans. So இதெல்லாமே வந்து database available ஆகக்கூடியது. So what bioinformatics can do? So bioinformatics என்னெல்லாம் பண்ணும், எதுக்கெல்லாம் அப்ளை பண்ணலாம்? So the bioinformatics, it's the bridge of the 3D structure based on the linear genomic information, the study of structural genomics. ஓகேவா, ஒரு ஜீனோமோட ஸ்ட்ரக்சர் எப்படி இருக்கும் அப்படிங்கிறது can't able to see our laboratory methods. ஸோ நம்மளுடைய லேப்ல வந்து நம்ம டிஎன்ஏ ஐசோலேட் பண்றோம், அந்த டிஎன்ஏ ஒரு ஜீன் ஆம்பிளிஃபை பண்றோம், அந்த ஜீனை வந்து we can't able to see. ஸோ அந்த ஜீன் எப்படி இருக்கும், we can able to find the banding using the agarose gel. ஒரு பேண்டிங் அதான் தெரியும், ஒரு fluorescent band கிடைக்கும், அவ்வளவுதான். So we want to structurally we can able to find based on the bioinformatics tools. So next one is gene expression analysis, the prediction of gene function and establishment of gene libraries. So it's called the functional genomics. Functional genomics. So how the genes are expressed. So நம்மளுடைய genes கிட்டத்தட்ட வந்து 1.4 billion proteins இருக்கு. அந்த proteins எல்லாம் வந்து எப்படி வந்து எக்ஸ்பிரஷன் ஆகுது. So how much amount of protein it will be expressed. So how the reaction is happening. ஓகேவா, ஸோ ஒரு கோவிட் வைரஸ் இருக்கு, கோவிட் வைரஸ் வந்து எங்க போய் எந்த ஜீனோம்ல வந்து அஃபெக்ட் ஆகுது. ஸோ வாட் கைண்ட் ஆஃப் ப்ரோட்டீன் இட் வில் பி ப்ரொடியூஸ்ட் ஆஃப்டர் அஃபெக்டிங் தி கோவிட் இன்ஃபெக்ஷன். ஓகே, ஸோ வாட் கைண்ட் ஆஃப் ப்ரோட்டீன் இட் வில் எக்ஸ்பிரஸ். ஸோ பர்டிகுலர் ஜீன் ஆஃப் இன்ட்ரெஸ்ட் அப்படிங்கிறது we can able to predict the gene expression analysis. So the next one is the ability to use genome sequence to identify proteins and their function, protein interaction, modification, and functions. So that's we call the proteomics. So proteomics. So genomics அப்படிங்கிறது வந்து it's the entire study of the genetic structures, genes. ஜீனை பத்தின படிக்கிறது எல்லாமே வந்து it's a genomics. அதுல gene expression எல்லாமே வரும். That's called functional genomics. Functional genomics. So proteomics is a genome sequence to identify the protein functions. So protein order functions, protein interactions, or protein inner protein, how they are interacted mutually, then whether the modified protein functions is எல்லாமே வந்து can able to study in proteomics. So and then is elucidating the function of molecules based on the structure. So structural based functions, molecules. So ஒரு molecules ஓட structural based functions. Then simulating the metabolism from the biochemical functions of an organism. Biochemical functions. Then molecular modeling and are the methods to predict the structure from function. Several methods of machine learning are used to predict the function from the sequence structure. Okay. So nowadays the medical science is blooming for the uh, discovery.
So your voice breaking, sir.
Discovery. So இப்போலாம் வந்து பாத்தீங்கன்னா so டைரக்டா வந்து the people okay இப்போ எப்படி கேயா
Sir, your voice is breaking.
வாய்ஸ் பிரேக் ஆகுதா
Yes, sir.
ஓகே ஸோ இன்னைக்கு ஒரு நாளைக்கு அட்ஜஸ்ட் பண்ணுங்க ஸோ டுமாரோ வில் solve the issues. Okay, I think system some issues in volume adjustment. Okay, so next thing the medical science would need to know the pathway to know which genome change could give rise to each known inherited disease, identification of gene causing disease, and also genetic therapies that can reverse disease phenotypes. Then data obtained from the functional genomics, proteomics would be using drug designing and drug discovery. So இது எல்லாமே வந்து we can use for the bioinformatics. Okay. The importance of databases, the more importance to the sophisticated communication network in biological and biomedical research. Okay, the ability to use the different online accessible software in molecular biology is becoming a mandatory for the all biomedical scientists. The current quest to sequence all the genes and to make the information available in the database, such that all the biological investigation must start with the browsing the data bank, making computer literacy compulsory for the all biology. The knowledge base arising from the genomics and proteomics research call for the well-planned bioinformatics program. So the database based on we can able to design our research process. Okay, without database, we can't be able to design our research because we need a diagram of the previous data. Then only we can able to include the persons. Okay, who's having find for example, so for lung cancer study பண்ணோம். So lung cancer study பண்ணணும் அப்படின்னா எப்படி ஸ்டடி பண்ணணும்? ஸோ எந்த மாதிரி செல்ஸ் எடுத்து ஸ்டடி பண்ணணும்? ஸோ எந்த மாதிரி ட்ரக் யூஸ் பண்ணணும்? So these are the things we can get it from the databases. Okay, ஒரு NCBA PubMedலயோ so nucleotide databaseலயோ வந்து we can get the information from the databases. So database இருந்து கனெக்ட் பண்ணி தான் we can able to find the solutions. Okay, the make a research problem statement. Okay, then give objectives. So the database is the more important. So one-time Gilbert is a base sequence in nucleic acids. இப்படிதான் வந்து ஃபர்ஸ்ட் வந்து அந்த பேஸ் சீக்வன்ஸ் ATGC அப்படிங்கிற சீக்வன்ஸ் டிட்டர்மினேஷன்ஸ் வந்து ஃபைண்ட் அவுட் பண்ணாங்க. ஸோ இஸ் a physics person. Is a physics person. Okay, he is interested in the theoretical physics. In his field converted to the molecular biology, genetic engineering. So based on his invention, he is Nobel Prize in chemistry in 1980. So after that only the people are finding the polymerase chain reaction. Polymerase chain reaction after invention of the prediction of nucleotides. Prediction of the nucleotides. The people are identifying the instruments we call the thermocycler. So Walter Gilbert. So today what we have seen. So just introduction about the bioinformatics. So bioinformatics is nothing but it's a biological information retrieved from the databases based on the structural molecules. So this bioinformatics we can deviate it in different ways. Is genome based analysis the genomic, then transcription based analysis we can find the transcriptome, protein based proteome, metabolism based prediction tools we can say the metabolome, the metabolites. These are the things, the different areas available in bioinformatics. The main targeting of the bioinformatics is the human genome project. So the successful completion of the human genome project in the April 2003. The main targeting of the human genome project is the genetic map of man, define the restriction fragment length, polymorphic character of individuals, then complete sequence of the complementary cDNA, then identification of the gene causing the genetic disorders. These are the objectives of the invention of human genome project. So totally 3 into 10 to the power of 9 bases available in the human genes. So after completion of the human genome project, the other genome projects, chemo plus influenza, Saccharomyces, Drosophila, Plasmodium, and some other plants, mammals, rodents, insects, bacteria, they have completed the entire genome. So first the database created by the Dehas in the macromolecules using the protein structure based. He designed the databases like GenBank, EMBL, PAR, Syscode. In future topic, we can able to discuss about this databases. What is the importance of the bioinformatics? What it can do for the bioinformatics? It's a prediction of the 3D structure based on linear genomic information. It's called the structural genomics. And gene expression analysis, the functional genomics. Then genome sequences, protein interaction, protein modification. It's called the proteomic studies. Then functional molecules based on the structure. Then metabolism of the biochemical functions of the organism. Molecular modeling and molecular dynamic method of predicting structure and function. It's a machine learning process. And major applications in the medical science, we need to analyze the pathways and find the inherited disease. So identification of gene causing the disease and also genetic therapies can reverse disease phenotype. Then data obtained from the functional genomics and proteomics using the drug discovery and drug designing process. So several importance of databases, bioinformatics databases, we can get a data from the whatever needs. Based on them, we can recover from the databases in different types, and information getting from the different country-based research, data bank, literature, review literatures, research articles, protein structure, DNA sequencing, nucleotides. So everything we can get it from the databases. Thank you. So complete the quiz. Feedback போட்டுருங்க. ரெகுலரா தான் சொல்ற மாதிரி தான்.